The year is 1953. James Watson and Francis Crick have just discovered the double helix nature of DNA. While this is a momentous answer to questions of the time, it gave rise to one more mystery. How does double helix DNA replicate? I welcome you to the great scientific debate, Meselson and Stahl's confirmation of the semi-conservative DNA replication model. Now, come on into my lab. While DNA was first discovered a staggering 153 years ago in 1869 by Frederick Miescher, the contemporary understanding of DNA was first proposed by scientists James Watson and Francis Crick in 1953. I'll bring up my whiteboard and we can take a closer look together. These men proposed the idea that DNA existed as two helical chains coiled around the same axis. Each chain was to contain a repeating structure of nitrogenous purine and pyrimidine bases that hydrogen bonded to an associated base on a second parallel chain. Watson and Crick further added to their namesake within the field by proposing the semi-conservative DNA replication model that Matthew Meselson and Francis Stahl proved in their experiment in 1957. During the 1950s, contention surrounded the mechanism in which DNA replicated. First, in 1953, Watson and Crick proposed the semi-conservative DNA replication model. In response, a scientific debate was sparked for all the fame and glory. In 1954, Max Delbrück suggested the dispersive DNA replication model in which all progenies would be segmented hybrids of daughter and parental DNA. In 1955, David Bloch proposed a conservative DNA replication model in which all parent DNA strands are never separated but still act as templates for daughter DNA synthesis. For Meselson and Stahl, the likelihood of the semi-conservative mechanism prevailed, and this became the focus of their experimentation from 1956 to 1957. The defining characteristics of the semi-conservative model are that two helical strands were to unwind from the coil and act as templates for the synthesis of new complementary daughter strands. The result is two DNA molecules, both with one intact DNA strand from the parent and one newly synthesized daughter strand. This pattern would continue. To prove this, Meselson and Stahl began growing E. coli on a plate supplemented with ammonium chloride containing N15, a heavier isotope of nitrogen. Thus, as E. coli grew, the nitrogenous bases of all parental DNA would contain the heavier N15. When sufficient colonies had accumulated, samples were taken. Ammonium chloride containing N14 was then supplemented into cultures in a tenfold excess to ammonium chloride containing N15. The cultures were then allowed to continue growing and samples were taken in intervals of several bacterial generations. All daughter DNA molecules of E. coli would be lighter due to the N14-based nucleotides. To observe the anticipated weight differentiation of parent and daughter DNA molecules, Meselson and Stahl utilized a separation technique, density gradient centrifugation. For density gradient centrifugation, a small amount of DNA in a solution of cesium chloride is centrifuged. Cesium chloride, when centrifuged, creates a density gradient. The heavier cesium metal particles experience greater centrifugal force, leaving the lighter salt particles close to the rotor center. As solutions are centrifuged, heavier DNA molecules will experience greater centrifugal force and find a sedimentation equilibrium closer to cesium, compared to the lighter N14 DNA molecules that will find equilibrium at lower densities close to the chloride ions. When the initial cultures were removed, containing only E. coli DNA with N15, the results of the density gradient centrifugation displayed only one density group towards the end away from the rotor. At the first generation, again, there was only one density gradient group. However, it was closer to the rotor center than the previous group. And finally, results after generation 2 showed two groups. One was identical to the first generation results, and the other was closer to the rotor center of the centrifuge again. It was known that generation 0, the parental DNA, would be the heaviest and thus the furthest from the rotor center. However, the comparison between generation 1 of the results and what Bloch expected disproves the conservative replication model. If Bloch's model were true, instead of seeing one density gradient band representing hybrid of parental and daughter DNA, two bands would be present, one representing pure parental DNA and the other representing pure daughter DNA. This was not represented in results obtained. Although this left Delbrück's proposal of the dispersive DNA model, the heat denaturation of hybrid molecules was conducted to weaken the hydrogen bonds between DNA strands. These samples were density gradient centrifuged, and two density bands were the result. One band was the parental N15 molecule, and the other was the daughter N14 molecule. If Delbrück's model was accurate, one band would be present representing the equal density of both dispersive strands. Meselson and Stahl's confirmation of Watson and Crick's semi-conservative model opened the doors for scientists to properly explain the mechanisms of DNA replication. Now, due to this revolutionary proposal and proof, researchers can finally understand and explain the basis of genetic inheritance and disease. Meselson and Stahl undeniably resolved the great scientific debate.